Without further ado, I'd like to introduce Eric Langer. He's a partner with the BioPlan Associates. Thank you.
Thank you, Brian. Appreciate that. Thank you all for coming, and thank you for Brian for setting this up originally. The reason I wanted to not wait just for some chairs is because this panel here is, as soon as I started to hear the kinds of things that they were going to be talking about, I realized that there's no way we could cover everything any one of them knows in 90 minutes. So this is going to be a tight squeeze to get all this great information in, and I don't want to waste a second of it. In fact, I don't even want to be up here talking. So let me start moving on real quick. You know what this is about. This is about what stainless and single-use is. That decision process is really challenging. In fact, there are courses being taught literally this month on how that decision process can be made. So we have with us some of the brain trust of this industry that makes this kind of a decision every day. So you don't even have to attend a course to be able to get that level of information. What I'd like to do with this expert panel is, the way I set it up was I wanted to be sort of a Dr. Phil kind of an experience here, where we've got the CMOs here saying this is the right way to do it. We've got over here experts in the industry. These are true biopharma experts that have made these decisions. And at the end, tying us up, is Sue Behrens. She has both industry and consulting experience in putting the engineering parts of these two together. So she'll moderate the session for us in that regard. But let me go ahead and introduce our speakers. They'll be presenting in this order. Actually, the slide is slightly mixed. Bill Hartzel is a Director of Strategic Execution at Catalent Pharma Solutions. He understands the world dynamics associated with this, and he and his company have made some very serious decisions on how single-use and stainless steel, the economics of that play out, and he'll share some of that with us. We'll have next following, we've got Stephen Perry. He's the VP of Technical Operations at Cook Pharmaca, where he has been making single-use systems work for his clients, and the operational decisions associated with that and the criticality of that. We're then going to switch over to the end-user side, and we have with us Joanna Pezzini. She's bioprocess engineering at MedImmune. Her background includes Biogen, IDEC, and Lonza. So she's seen this from both sides. So I think that her perspectives on how it's done in downstream, and she could talk from the full process, but we're going to break that down into just downstream from Joanna. She's got 3 case studies for monoclonals that she'll be able to share with us. Dani Belen is the Senior Director of Global Technology Innovation at Sanofi Pasteur, and he'll be talking about how stainless steel and the single-use decisions are made in real life. And at the end of the presentation, we have Sue Behrens. She's Senior Director of Process Technology at IPS. She's going to provide us with consulting's best advice on stainless steel versus single-use. Her background includes, of course, Merck and other very significant experience as well in these areas. So if it's all right, if you all don't mind, we'll do a quick pause when the chairs come, but I'd really like to get started. Bill, can we have you come on up? Perfect. Thank you so much.
Excellent. Well, I have to admit, this is the first time I'm giving a presentation that somebody's actually referred to themselves as Dr. Phil. So I'm hoping I'm not going to go through some, psychoanalysis. But I do appreciate everyone and the opportunity to come speak, today, and especially, Catalent has a very unique situation in where and how we came about making the decision to come to single use. And I think at the beginning, one of the big things that you have to focus on from a fundamental standpoint is really understand what are the goals and objectives that you're trying to accomplish with either your build or your manufacturing process and what you need to deliver. And I know that those goals and objectives are going to vary greatly between innovator companies and companies in general or service providers like we are. And so one of our big looks when we looked at going from a single-use technology or going from stainless steel to single-use was really how does that fit and what's that philosophy around our business models, and then how do we align really our business and our manufacturing strategies together. So for us, one of our main focuses is how do we provide flexibility to our customers in multi-product facility, and also how do we drive speed. So from our standpoint, we have a cell expression technology that it's based on how fast we can get products to clinic. And how do we do that? So the cell expression technology, how do we link that manufacturing strategy together to be able to provide, and be a catalyst for our customers, as they're coming to us, and how do we get them to clinic faster? And so this is where we'll start walking through on the presentation standpoint, really how we embraced that decision. So just from a history standpoint of the site, really want to highlight a couple things. We've gone through 3 major expansions since the early 90s. We actually were a transgenic organization, and that's what we were focused from a technology standpoint. So we actually went from the manufacturing inside of a cow into a stainless steel reactor. So for us to make the decision to jump from a stainless steel reactor into a single use wasn't as large as we had already made previously with our technology. And Catalent was at another really unique situation. We knew that we had to expand. We knew that we had to expand back in 2008. The market started to crash, so we put the hold on the project. But at that period of time, we had already gone down and tried to order our long lead time items. So we had bought 6 stainless steel reactors at this period of time. And then in 2011, the project was reengaged, the market started opening up, we needed to keep growing, right? So we needed to then go through the build, and I'll talk through that decision process of why we ended up and where we ended up. So again, we talked about what our focus and our goals were. And our focus and goals was really, from a site standpoint, it was about growing. We needed to grow that facility and grow that market. So we had an expansion plan and process that we were going through. And it was really focused on the phase 1, phase 2 area of manufacturing. Also what we wanted to look at was even a growth, to go away from just not only our cell expression technology, but to use other cell expression technologies. So we had to be able to encompass that in our design philosophy and in our drive. And not only was it about getting bigger, but it was also about getting smaller, that we were able to do smaller, more efficient turnaround. And those types of things were at the focus of our manufacturing strategy. So again, we went through and we looked and we started to weigh out the options. And this is, coming from the industry, of their looks and overview. Right, there's energy savings, labor savings, and consumable products. We're going to go, or energy savings on the labor side, much better in situation for the single-use side. Consumables were going to go up. From a contamination rate now, we haven't seen this. This is from the industry. We haven't had those issues, but there's an increase on that side. But really, one of the things that we were looking at is really driving that flexibility, especially going into a multi-product facility. So really, when it comes to us, this is where, from an evolution of thinking standpoint, where it happened in 2011, we basically reached out to an engineering firm, and they did an analysis for us. And they looked and said, okay, even though you've had all of the investment you've already made in your stainless steel reactors, even though all of that is already there, the capital that it would take for infrastructure and timing of all of the support systems actually made the decision CapEx neutral. So from our standpoint, we were able to look and say, okay, well, what should we do? And there was the labor savings that we would get. It de-risked our build because we were able to take out time. So going to a single-use process and the single-use manufacturing reduced a lot of the risk that was associated with the build itself. And that was a big driver from our decision-making process, as well as our ability to then leverage and align, both our manufacturing strategy, with our business plan and our business models to the market. So really, our big moment of truth came in when we actually sold those stainless steel reactors, to our competitors. And so from that standpoint, that's really where we embraced it. And really what we ended up leveraging, and really what it allowed us, again, going back to a faster turnaround. So from a build standpoint, this is one of the big things, that we were able to take a slab of concrete in the inside structure of a building, and we turned that area in 1 year into a GMP revenue producing facility. So from a build standpoint, this was one of the major gains that we got by going for single use. Not only that, but now we're able to really efficiently be able to handle a multi-product. Because the systems are closed, And because you're able to handle that, even through that configuration, we're able to handle 9 different products at one period of time, and we're able to control that. And that's the only way we could do that is through single-use operations. Again, we're able to do it then from a fraction of the cost of what a stainless steel or fixed system would have ended up costing. So, again, we were very excited about the project, turning it around, and I just wanted to really close and highlight, from a transitioning standpoint, some of the big lessons learned that we had going from single-use to multi-dose. And the points to consider for us, especially being a service provider, was transitioning our customers, the customers that are already done things in single-use reactors. And one of the things I'll really highlight is the fact of working very close with your supplier base and really looking and treating them as a partner in how we transition these things forward. That was a tremendous advantage for us when we were going through, because we were able to get a loan from our sub-manufacturer, our bioreactor, and we were able to then we were able to do those through scale, and we were able to work with our customers to move that through. Ultimately, you're outsourcing the validation. So you're not buying bags, we're not buying tubes, what we're buying is validation packages. If we wanted to buy a bag, we'd go to Doritos or Frito-Lay. For us, we're buying a validation package that we're getting, and we have to outsource that. So from an organization, we have to be comfortable, but that's where we really need to leverage that supplier relationship. Again, the criticality of supply chain, from change control and inventory management. From our standpoint, it was training as well that we needed to get there. And then ultimately, it's solid communication internally, training our internal folks, how we're doing, at what phases we're at, and also in the external community, both internal to our business as well as our customers, and making sure that we have good communication with some of those keys that we had from transitioning from a a stainless steel facility into a single-use manufacturing facility. So thank you. So now I'll leave it up to you, Steve.
I'll tell you what we're going to do. We're not going to let them off the hook quite this easily.
Nice.
Rather than a bunch of talking heads going out at it, what I want to do is start this presentation with a little bit of interaction. So I'd like to open the floor to your questions. I'll take 1 or 2 real quick questions that we'll then be able to answer, and we'll do this with each of them. And if any of our panelists have any comments on that, I'd like them to chime in as well. Do we have one question? I'll tell you, I've got a question in back, and we'll repeat the question if you can share it with us.
This was not the essential of a validation package. So, and I can get more with our quality department, but when we're talking about a validation package, it's really along the lines of your change control processes, how you have from your supplier and your supplier auditing, right? Those are the things that we need to know and understand from a organization standpoint, that your processes that you go into making that, if it's a bag, if it's a tubing set, that you have the right level of controls. So when we're getting materials, that we are confident that we are getting the same thing on a repeatable basis.
Right. And I've got one quick question for Bill as well. What single point, if you were buttonholed by an end user, what single point would you share with an end user, a biopharma end user, if they were to make that same decision on making either the transition to from stainless to single use, or decision between one or the other? What single point would you recommend they consider?
So again, I think that's a tough question, because it really goes back to what you're trying to focus on what are your goals that you're trying to achieve. So if you're in the later stages and from an end-use perspective that you're going to be running something consistently, is much different than if you're in early phases and you don't know the risk of a multi-product, right? So do you want to build, and do you have all of this fixed asset for something that you know? And if you're in later stages of a build or a process, you may want to take that and go with it. For being somebody that's early stage, so if you're looking at it, the flexibility and the elimination of cross-contamination and cross-contamination potential, I still think is one of the main drivers. You don't have to clean. You can get rid of that, and you have dedicated equipment per run, per batch, for every product that you're doing. And I think that's a really key point that single use provides that a multi-dose or a fixed facility won't have.
Okay, thank you very much. Thank you very much for that.
Okay, thank you, Eric. I see the chairs have arrived, so feel free to take a seat. I'm glad I'm speaking to a standing room only crowd. This is great. So I guess, should I go ahead and I'll just keep going?
Yeah, keep going.
Okay, I guess I'll go ahead and keep talking and feel free to take a seat if you're standing. Can you hear me, by the way? Oh, there, it's a little bit better. So I'll be talking about stainless steel versus, obviously, single-use. I'm with Cook Pharmaco. Let me tell you a little bit about Cook Pharmaco. For those of you who are not familiar with the company, we were established in 2004, and we're a division of Cook Medical. Cook Medical is the largest privately held medical device manufacturer in the world. We just celebrated our 50th anniversary a couple years ago, and Cook Pharmaco just celebrated our 10th anniversary. Cook has a long history with contract manufacturing. Back in the 90s, we had a drug product contract manufacturing operation. We sold that operation for various reasons, but got back into the business again in 2004. Initially starting with drug substance, and you see there we have the 2,500-liter train. By the way, we have our campus is huge. We have almost 1 million square feet under roof. 2 very large buildings. Used to be the largest television assembly plant in the world. That was gutted, shut down, gutted, and we've made it into a state-of-the-art biopharmaceutical manufacturing facility. In addition, so we have plenty of room for expansion, potentially to larger scale, 5,000, potentially even 10,000, or possibly even larger. We have plenty of room there, and we just need to figure out the right time to do that. A drug product, we have a syringe, vial, lyophilization, packaging. I can't really do justice. I've got a few photos in here, but I really can't do justice to our facility. You really have to come out and see it, or please stop by our booth. You can't miss the big sign back there with Cook Pharmaco. We also have very good development labs, process development, analytical, and formulation. So we really are a full-service contract development and manufacturing organization. We call ourselves one source, one location, everything from vial to vial, in addition to the packaging operation. So I want to talk to you a little bit about single-use and where we use single-use at Cook Pharmaco, and try to kind of rationalize where we use single-use and where we use traditional stainless steel, reusable components. So where do we use single-use? So buffer, media prep, as well as this post-filtration. So we prepare all our buffers and media ahead of time, sterile filter it, and then that is then used as needed by the operation. Of course, our seed train, mostly shake flasks, is obviously single-use, and the seed bioreactors up to the 200-liter scales. We have wave and stir tank single-use bioreactors as part of our seed train. Getting to the downstream part of the process, any in-process mixing, for example, low pH hold, acid, viral inactivation, conductivity adjustments, any mixing will be single-use. Any in-process storage, as we move through our downstream purification, is in single-use, up to 1,000 liters we can handle. And finally, any filtration will be single-use. That's depth filtration, virus filtration, or sterile filtration. We have no stainless steel filter housings in our plant. All of our filters are either capsule filters or pods, which are modules, which are assembled into an assembly and held in a housing. So really, everything filtration is single use. So now, where do we use stainless steel and reusable? The production reactors and in this case the N-1 reactors. So our 600 liter and our 2,500 liter reactors are single use, and part of that was if you think when we were built in 2004, really we single use bioreactors were still fairly new back then and limited to fairly small scale. So that was part of the rationale for going to stainless steel there. Centrifugation, harvest tanks as well, size to fit our bioreactors, our chromatography systems, and our ultrafiltration, diafiltration. So those are your traditional stainless steel reusable components. So we clean them, sanitize them in between use. So kind of where, again, to try to rationalize why we have certain operations are single use and certain operations are reusable. So I think you've probably the ones that are talked about the most are, avoiding the cleaning, validation, or verification. It's single use, you throw it away. Rapid changeover between runs. Also, again, the flexibility, which was just referred to earlier, to accommodate multiple processes. Those are very well-known rationales and reasons for using single use. But I want to talk a little bit about some other more practical uses and reasons really related to operations. And one is really scheduled decoupling. So I mentioned earlier media and buffer prep. We accommodate a number of different processes, again, as a contract manufacturer. Some of them use a great deal of different buffers and medias. Others are fairly more straightforward processes, so there are not that many buffers and media to be made. Having the single use and decoupling the buffer and media prep operation from the actual processing operation allows us to level out our labor, and so we can schedule those buffer and media prep operations well in advance of when those materials are actually needed. If you can imagine, if we tried to build a train of stainless steel hold tanks and so forth, many of them would be idle or many of them would be unused for different processes. So this is a very good way for us to address the flexibility we need as a contract manufacturing organization. And then the other piece is risk mitigation. So our philosophy toward making buffers and media is we'll prepare the buffer, sterile filter it into a separate pre-sterilized bag, and then that will be held until needed. That will be stored under the appropriate conditions and held until needed. So that's particularly useful for example, a bioreactor operation as an alternative to preparing the buffer media in a separate tank and then filtering it directly into the bioreactor. You can see at that point you're coupling your two operations, and you also don't have the opportunity to mitigate the risk. So we're able to do bioburden testing or whatever we need to do on the buffers or media they're going to be used in the operation. So decoupling that allows us to, again, level out the schedule as well as put in some risk mitigation in case there were something wrong with the buffer or media. We would not be committing it to the process. So that's another advantage of using single use. But I want to talk a little bit about the challenges because these don't get talked about too much. I think some of that was mentioned as well. Inventory management and supply chain. So typically, once you get into it, if you want to implement a single-use strategy in an operation, you're almost certainly going to be dealing with some type of custom assembly or a custom bag or bag. Again, it's been customized in some way. And again, so the importance of working with your vendor is going to be important. But also from a supply chain point of view, again, as a contract manufacturer, if we have too many different configurations and so forth, and we have to deal with lead time issues, which can be an issue a lot of times with these single-use components, you can imagine we then get into a situation where we have very high inventory costs. And so managing your supply chain and managing your inventory and managing the different configurations you have has been very important for us. So we have to keep control over that and provide the flexibility we need, but also provide the control we need over the cost and the configurations. Obviously, vendor quality is important. You're really relying on your vendor now. You're really outsourcing part of that, of the assembly process. And so I think it was mentioned to work with your vendor as a partner. It is very important that you consider your supplier as a partner. You work very well with them. They have a good quality system. Of course, we audit vendors and make sure they have the right quality systems and so forth in place. One thing that's important, if you talk about quality, is that whereas typically if you have a bag or so forth with connections on it, we need to make sure that the vendor typically they'll test out the bag and make sure the bag doesn't leak, but also just make sure the connections and so forth. So there's a lot of detail you get into when you're implementing disposables that we've learned you really have to pay attention to. Design limitations. Again, if you've got a bag or a single-use reactor or so forth, what's your flow out of that bag? A lot of times we'll find that we can accommodate the volume of the bag, but we're limited on tubing size at the outlet or on the inlet. So you could collapse an inlet tubing, or you could overpressurize an outlet tubing. So again, a lot of operational details we found we have to think through. Setup challenges as well, and we'll get into actually working with the operators. So it's very important that the operators are very well versed and have had a lot of practice with these systems. If you don't set up a bag correctly in a single-use mixer, for example, you can get bag folding, you can get things like kink tubing and so forth. All these things are very important, and with, again, a lot of new types of connections coming out and single-use components coming out, it's very important that those operators really have practiced it and that they've really been tested. Finally, I've kind of skipped forward to the operator training, but that really goes hand in hand with that, with a number of new aseptic connectors. Some of them are very technique, reliant on technique, and you're really relying on your operator to make an aseptic connection correctly, and you could be introducing some risk into the process if the operator's not trained or doesn't have enough practice. And finally, the impact on the process. Again, that gets into any impact that the film may have on the process or any of the components that are going to be contacting the material. That can particularly be an issue sometimes in upstream processes where there may be some impact on the cell growth. We've seen that in the past and have had to work around that on occasion. But that's typically where that issue will manifest itself. So again, very important in small scale and process development to work with a scale-down model of your disposable system before you implement it in large scale. So to conclude, what is our approach? So we use single-use technology where it's well established, it's low risk, and really it confers an operational advantage. We are continually evaluating single-use applications, looking at potentially moving to some larger single-use bioreactors. But again, that's going to take a lot of work and a lot of evaluation. But we have seen that the single-use bioreactor sizes have increased since we built the plant, obviously in 2004. And finally, operational details, again, are very important. You can't overlook that. Practice, trials, engineering tests, operator training, very important when implementing a single-use system. So that's it. Thank you.
Thank you very much. Thank you very much. We looking for a question from the group for Steve on implementation of a hybrid system at Cook Pharmica.
Okay. Yeah. So the question was, why did we go with single use on some equipment and traditional reusable on the other equipment? We're always looking. Again, I think it was also mentioned. What are we trying to accomplish here? Obviously, we want to accomplish a great deal of. As a CMO, we need to have a flexibility, but it's also important that we have reliability for our customers. That's very important. If a batch fails, the customer doesn't care whether it was single use or traditional. They just care that we have to reschedule a batch. So that's very important. So we want to use single use where it gives us a flexibility, but we're somewhat risk averse, I would say, because you're only as good as your last customer, right? And so it's very important for us to be very sure before we jump into a single use. So again, where we've chosen to stay with traditional has been really where there are not that many single use offerings. Centrifugation, or chromatography columns. Large scale chromatography columns, chromatography systems. There are some systems out there, and of course we take a look at them, but we're not kind of ready to make the leap for those. I think the next place we probably would implement the next step of disposal would be looking at larger single-use bioreactors. But it's really, I guess, the balance of flexibility against risk for the customer. Thank you.
Can I ask you the same question?
Sure. Yeah.
Okay, I going to ask him the same question. If you had one piece of advice to share with somebody making that same decision on stainless steel versus single use, what would that single piece of advice be?
Yeah, I think is just be very clear on what you're trying to accomplish, and I think that may have been a similar answer, but I would say the same thing. Why are you implementing disposable single use? Is it because that seems to be the way the industry is going? It's important to be looking at new technology, but I would say we haven't. It hasn't really been. It's the rationale for doing that has to be from the operational side. So if there's a good reason to implement single-use technology because it gives you flexibility, or you have a facility that kind of drives you towards single-use technology, then that's fine. But just make sure you know why you're doing it, and don't underestimate the challenges in implementing it.
Thanks very much. Thank you. Thank you. I'd like to introduce Joanna Pezzini. She's a MedImmune engineer. She's had experience in development of skids, especially related to downstream, and her experience, I think, is going to be very relevant because this industry has been looking for downstream single-use opportunities. So we look forward to understanding case studies based on what MedImmune has done.
Okay, good afternoon. Again, I'm Joanna Pezzini, and I work in the BioProcess engineering group at MedImmune. And our group does a lot of technology transfer work, and so I've had the opportunity to work on several projects implementing UFDF skids at various different facilities within MedImmune. So we have been looking at a single-use UFDF skid at a 500-liter GMP facility, a 2,000-liter GMP facility, and then also at a 500-liter pilot facility. And these facilities are slightly different. The GMP ones are primarily stainless steel facilities that we're looking to implement single-use in various situations. And then the non-GMP pilot facility is much more single-use in general. We have some single-use bioreactors, some stainless. We primarily use bags for buffers and a lot of tubing connections. So I'm going to review some of the drivers behind the decision on purchasing these single-use skids, some of the cost benefits and time savings that we were able to achieve or not achieve, and then also go into some of the technology challenges that we face and are also still working through. So some of the primary drivers for purchasing a single-use UFDF skid, the biggest one was really the larger operating range of the single-use skid. So it has a much smaller hold-up volume than we typically see with the stainless steel ones, or at least the stainless steel ones that we had quoted to us. And this allows us to go to much higher concentrations or work with lower production, tighter batches. And so the skid also has multiple bag sizes and multiple size tubing kits that you can use with the same skid. So you can get a much larger minimum and maximum volume range and pump flow rates with that. And then the single-use skid also has a much lower capital cost. I'll go into a little bit detail in later slides in terms of exactly where it fell compared to a stainless steel system. And then we also found that the single-use system is very portable. So for a few different reasons. One is that it fits through a 40-inch door, and we're working with some small suites with some small doors, so this was actually pretty critical for us. It's also much lighter weight, so we can easily wheel it in and out of that room. And we found that we can also bring it between GMP and non-GMP facilities. So because it's single-use, we don't have any product contact parts that are moving between these GMP and non-GMP spaces. And again, I'll go into a little more detail on that and how it helped with some of the validation and commissioning work. So as I mentioned before, the capital costs are a lot less for the single-use skids. So we actually found that capital costs are about 1/2 to 2/3 the cost of the stainless steel system. But then, of course, with the disposable systems, you have all the consumable costs. Every time you want to run a batch, you need to get a new tubing kit with all the instrumentation and the pump heads and whatnot. And so we expect to break even with about 30 batches running on this single-use system. So depending on how many batches you run each year, that either is or is not very many batches. So it's not necessarily a cheaper system, but we do have more flexibility. We also looked into a cost analysis in terms of using the single-use membranes versus reusable membranes. So we really are standardized on a reusable membrane currently, and so we are still using that. But we also wanted to see, would it be cost-effective to use single-use membranes? And it turns out that if we use single-use membranes more than once and we clean our reusable membranes, then they're cheaper. So single-use membranes are just slightly more expensive than the disposable membrane, and that difference is kind of made up for with the cleaning cost, the hydroxide and the Wi-Fi and whatnot. And we also find that as we move up in scales, we get slightly lower break-even points, so fewer batches before your single-use and stainless steel systems become equivalent in cost. And a big cost savings that we did achieve was from validation. So that was very helpful. Validation cleaning we didn't need. We also saved a lot of time with commissioning in the actual GMP suite. Because we can move this suite to the pilot facility, we did all of our commissioning and testing and automation work at the pilot facility. So this was very helpful in terms of downtime in the manufacturing facility. The single-use TF system also, when we're actually running it during normal processing, we find it takes more time to set up than the stainless steel system because that already has the filters installed. They're stored in hydroxide. We just have to clean it. And because we're still using reusable membranes, we still have to clean our system beforehand because we haven't found a suitable reusable or single-use membrane. But when we find a suitable single-use membrane and we manage to validate that in our process, then we expect the setup time to reduce by about half. So it'll take about 2 hours, 2 people to set up and run, or just the setup and then running time on top of that. And in general, when we were operating the equipment, we found that operation was very easy. The automation ran very smoothly. It was basically just a setup and let it run. And this was a big difference for us at the 500-liter GMP facility, because previously we had a very manual setup, and this was one of the drivers to purchase a new skid, was to get an automated system. And it really did achieve that point. So I'm going to go into some of the challenges that we found with this single-use system. And these are specific to the UFDF skid, but I think that they also apply to a lot of single-use equipment in general. So for us at this UFDF step, it was new to need an extractable and leachable assessment when we're moving from the stainless steel. So that was something that had to be considered. That was extra time to cover. We also had to have a new instrument calibration approach. So we are relying primarily on vendor calibrations and certificates for the instruments that are on this UFDF system, which we hadn't done previously. And we also need a new post-use calibration approach. So we don't do post-calibration on all of the instruments, but we are still doing them on the very critical ones, like the pressure indicators. And we'll have to see how that moves forward based on the data that we're collecting over time. So we're also, one other concern that's come up is the reliability of the kits. It hasn't been perfect, but it's been pretty good. We have lower pressure ratings on a reusable system than a stainless steel system. And so if we're operating at our pressure limits, typically on the stainless steel system, we find that we have to run at slightly lower flow rates. And this isn't across the board all the time, but in a few instances it might come up and make this process step a little bit slower. And then another issue has been the accuracy of the turbine flow meters. So we would typically use the turbine flow meters to calculate our diafiltration volume. And these flow meters currently aren't accurate enough to allow us to do this. And so we've had to move to a floor scale to be able to collect that volume and accurately measure it. And we're not currently aware of a good disposable flow meter that could be a replacement for this. So if anybody knows of one, let us know. And then the last point that I wanted to bring up for some of the challenges that we've been facing is our mixing in these in the retentate bags. So we've currently found that we only get good mixing up to a 20-liter bag, and so we can't operate with bags larger than that size currently. The bags rely on retentate flow alone for mixing, and when we get to the high concentrations, the viscosity gets too high and the flow rates get too low to be able to rely on that. So it was okay in the 20-liter bags and below, but not higher than that. So we are currently looking for larger mixing and different mixing systems to be able to use with the system at the 2,000-liter scale. We're covered at the 500-liter scale where we already have the system implemented, but at the 2,000-liter scale where we've pushed it and we're looking to work out some of these details, we still need to find a sufficient mixing system at that scale. So we're looking for active mixing with an impeller in a bag. And we're also looking at specific impellers because we have found that there are some shear issues with certain impeller designs. So in summary, we would say that the single-use skid does provide greater flexibility. We have found that we could get one skid instead of two skids at our 2,000-liter scale because of the flexibility that the single-use UFDF skid provided, and it can be moved in and out of these rooms very easily. However, the cost savings may or may not be there depending on how much you're using it. And we still do have some technology challenges, but I think we feel that we can get through all of them, and so we will get these systems working well.
Thank you very much. Joanna, I did have one quick question. I hope you have a question as well. Joanna has experience both on the end-user side as well as on the CMO side. I would suspect that CMOs have a different perspective on this calculus than end users. Can you talk to the differences between how a CMO may make a decision on something like this versus an end user, and how you'd recommend that process be considered?
Yeah, so I think maybe not necessarily a strong difference between the CMOs and biopharma at least, but I think it really depends. I've seen at different times different budgets are larger or smaller than others, and so the capital cost savings can either be more or less important, and I think that really depends on the budget cycles and how the economy is doing and whatnot. I think if a CMO is already established and you have a great cleaning system, you've already got that CIP system installed and the sterilization installed, it doesn't necessarily seem that there's a huge benefit in your cost reduction from going disposable. However, if you're starting up a new facility or you need the greater flexibility, especially in these pilot facilities or the more clinical facilities where you're dealing with a lot of products, a lot of different scales and volumes, then the disposable equipment becomes much more useful.
Question from the floor. Repeat the question.
So the question is around leachables and extractables and what we focused on. So this is, I think I don't have the exact information on what packets we use. I can get the primary contact for that afterwards if you're interested. Mm-hmm. Yep. So I think one of our big concerns is that some of the lead times seem to be creeping up, and so we're a bit concerned if we need to get parts quickly, that might not necessarily be able to happen. And then we've had just one issue in terms of a kit that we'd had to replace. Try to install it, it wouldn't install correctly, but there was an issue with it and had to take it out. But that was before we had product in it, so. Mm-hmm.
Okay, thanks very much. We'll have time for some Q&A at the end as well. Let's introduce Dani Belen from Sanofi. He'll share his experiences in single-use stainless decision making as well.
Okay. Good afternoon. So I took the perspective of talking about vaccines and our choice of single-use versus stainless steel based on the breadth and differences of all the different vaccine products that we make and that are made. And so this is from a little bit different view, but with similar kinds of critical decisions that we have to make at each step. Oops. All right. There we go. All right, so the obligatory statement that says that I'm not responsible for anything that I say, or my company's not. And I thought I'd start with just a brief history, in terms of what I'm going to present. But the first vaccine product that we can say that was scientifically or medically designed was a smallpox vaccine in the late 18th century. So we've been doing vaccines for a long time, and it's only recently that single use has come into play. I'll also say that as an industry, we're pretty conservative. A lot of our products most of our products go into healthy people and healthy babies. And so we're not trying to cure anything, we're trying to prevent disease. And so we have to be extra cautious. So we're a very conservative corner of biopharma. I started actually at a small biotech at the beginning of 2001, working on what is the nth generation of the smallpox vaccine. We'd gotten a contract from the U.S. government to build a new vaccine for the National Pharmaceutical Stockpile, and it fell on me to do the process development work, build a group, and so on. So the story begins and ends with me picking up the ball after Jenner way back when. Just a word to kind of lay the groundwork about all the different kinds of products that we work on. A little history of how the vaccine industry has evolved, the different kinds of products that are produced. Smallpox vaccine was a live attenuated virus. Of course, we didn't know a whole lot about it back then, but in a historical perspective, it's a live attenuated virus. We moved on and began to make toxoid vaccines, and the evolution continues. Now we have virus-like particles. We're starting to look seriously at DNA and RNA vaccines. So there's so much diversity in there that you can imagine that it's almost on a product-by-product perspective in terms of single-use versus stainless steel. So just a little bit of an overview, again, of the different kinds of antigens that we produce, the different kinds of vaccines, all the way from lipopolysaccharides and polysaccharides, typically in childhood and elderly vaccines to prevent pneumococcal disease, newer conjugates of those polysaccharides to make them more effective, viruses, which happens to be the area where I have the most expertise, both live, attenuated, and inactivated. Bacterial vaccines. At the beginning, this was killed whole cells, whole bacteria for pertussis, for example. Now we have DNA viruses. We have acellular pertussis vaccine where we purify 5 different antigens out of a bacterial culture and reformulate them to make the vaccine. We've tried peptides. We've tried a little bit of everything. But the biggest moneymaker is still the seasonal flu vaccine, at least for us and Merck and some of the other big companies. And where do we make that? We make that in eggs. Not really amenable to single use. So there's so much diversity in here, you really have to think about it on a case-by-case basis. So again, my expertise is in viruses and cell culture and viruses made on cultured cells. When we start to talk about all of the advantages, obvious advantage of going to single-use or a single-use dedicated facility is the initial CapEx. You're not buying all that stainless steel. You're not installing steam and CIP and all that stuff. You're building a clean room, almost stripped bare. Some utilities, gases, airflow, but you don't need all that stainless infrastructure and all that piping. And you don't have to validate it and clean it and continuously validate it. So there's an ongoing advantage to not having all of that built-in stuff. Facilities operations, there's always new challenges in terms of moving on to the next product or building the next product. We can't build two suites that are the same because everyone is custom designed for the particular product that we're working on. And as been mentioned before, when it was stainless steel, we were responsible for keeping it clean and keeping it validated. And now the product contact surface is coming from a vendor, and so we have to shift the responsibility to those vendors to give us the product contact service that we need. Leachables and extractables, particulates, all those fun things that we now have to worry about that we didn't really have to worry about before. Some vaccine products are more amenable to others to working in the single-use realm. We can grow cultured cells in single-use bioreactors. We can propagate the viruses in those. But we always end up with a hybrid system because there aren't good centrifuges and column chromatography systems that can work at the scale that we need with the speeds that we need. So we always end up with a hybrid system. Some stainless or CIP or SIP capable equipment commingled with wherever we can put in the single use. We can grow anaerobic bacteria because typically the mass transfer rates for anaerobic bacteria, so tetanus, other Clostridium type bacteria, they work fine because they're mixing and shear, and mass transfer is more like a mammalian cell and their demands. Seasonal influenza, we're in eggs. There's no way to make 150 million doses between June and October without using an egg-based infrastructure at this point. And finally, aerobic bacteria, E. coli or other aerobic bacteria that we would use, the mass transfer demands are too high for us to be able to work with single-use bioreactors and so on. So it really comes down to matching what your product needs, the biological system that you're working in, against what the different capabilities in these different areas look like. We've started to look at the concept of modular or mobile units. This is just one example. This company, GCON, affiliated with Texas A&M, where they have mobile suites, can be moved around, reconfigured, connected. It's almost like dominoes, building a little structure. Facilities operations are a real challenge. There's a different level of training required of operators rather than working with a large piece of stainless steel in a steam plant. You're now working with complicated, relatively fragile bags and tubing and manifolds and so on and so forth. You can end up with a lot of spaghetti on the floor. It's easy to tear or stress a bag, getting it out of its package and getting it open and put in place. It's a whole different realm for the operators to have to deal with this stuff. They're used to clanging around with tri-clamps and so on, and we're giving them a plastic bag with a lot of tubing that they have to carefully unfold and mount and manipulate. We spent a lot of time, we had a lot of learning, and we had to spend time learning the vendors about their packaging and how they present the product to us. If it comes packaged in a way that is too complicated for us to unwrap and set up, it's a big deal. We've also, in converting facilities, we've run into problems with storage space. The packaging has to be robust, and so you end up with lots of big cardboard boxes, and dealing with all that is a new challenge. We've had some issues, just to expand on that, operator, vendor kind of matrix. We've had to work with vendors and operators, putting them in the same room, figuring out what it is that the operator needs in order to be able to do what he needs to do, and then what the vendor needs to do to be able to make it easy for the operator to do it. And this is just an example where we were working with a bioreactor manufacturer, and it wasn't clear to us or them how robust different pieces had to be, and we had some failures. And they seemed to be intermittent. You'd have 2 or 3 runs that went well, and then all of a sudden the next run something would crash. We'd get a contamination. What's going on? Well, it turned out that some of our processes were kind of stretching the limit for what had been tested, and so we tore things up, not really recognizing where the weak points were. So just a few takeaways, and I listed them here. I could go on for hours. I spend a lot of time working with our quality systems and so on, so forth, and teaching. From a process engineering or process development perspective, and bringing them into the fold, if you will, as to why we're doing this and what the advantages are and what they need to worry about. But I can summarize this. The question that Eric asked earlier about what is the one piece that you might consider, I think it distills down for me, and my experience is that you need your quality and materials management folks in the room at the beginning. They need to understand your thinking, why you want to go this way, and they have a perspective in terms of validation and all that stuff that I might not have as a bioreactor biologist. So bring them into the room, get the conversation started early. It's no fun to get 6 months or a year into a development project and then have to go back and explain yourself to your quality or your materials management stuff. Why we chose this? What did we do? How do we go about thinking about this? So bring them in, get their perspective early on. And that all feeds into the idea of bringing them into the fold in terms of change control and the idea of flexible manufacturing and the pieces of equipment that are supportive, tubing welders and so on and so forth, that make the single use really possible. Really all that's changed is the dress code. We're still doing it the same way we were back then. Thank you very much.
Dani, thank you very much. I did have one quick question for Dani. You'd mentioned training a number of times and the issues associated with training. Can you comment briefly on what you might recommend as an appropriate training program to bring all these pieces and parts together so you don't end up with an ongoing disaster?
Yeah, it really is. We bring the vendors in to do training. I mean, I'm sure you guys do too. They know their product. They know how to deal with it. They designed it. And so we'll have a lunch and learn half a dozen times a year with a vendor when we bring in something new or when we're scaling up. Use the expertise that's out there and run programs that put the operators, the guys on the floor, in contact with the people who made the product. Don't try and be the intermediary. You'll be chasing your tail. So that's a big deal, bringing the outside expertise in.
Can we bring in one comment or question from the audience? To repeat the question as well.
Yeah, so the question relates to more manual operations relative to stainless steel. And this goes back to the training bit too. But a stainless steel, say a bioreactor or a bioreactor skid, is a fixed piece of equipment. And really all you're doing is changing what you're flowing through it at any one particular time. And it's all set up with valves, and now they're automated systems and so on. I want CIP, I push the CIP button, and it goes like that. With single use, I'm bringing in what is a more and more complicated piece of plastic engineering, tubing manifolds and and especially for upstream, and for some of our vaccines, they have to be aseptic from soup to nuts. It's really important that when it comes out of the packaging and gets set up and installed, that it remains integral. I mean, think of about a 2,000-liter bioreactor. I mean, that's a big bag. And there's tubing coming out here and there, and you can imagine what it looks like coming out of the package. And so it is a highly manual operation and technically demanding to get it put together right so that you can begin doing what you're going to do. And then at the end, breaking it all down and so on and so forth. So your operators need to be nimble and your SOPs need to be designed around that unpacking and installation piece. You can't just say, open the package and blow it up and away you go. How do I get rid of bioburden? In what context? I'm sorry. So I'm growing mammalian cells, and if I have any bioburden, I'm dead. I start over. Maybe somebody in the audience has a better idea. If I get bioburden, I start over. I'm growing viruses. I can't filter out. I have real constraints. So I start from a closed system or an aseptic processing perspective. I just don't want to give it a chance. But maybe if somebody else in the audience could have a conversation or has a better idea than that, please feel free. So the question, I think, is for the whole panel about failure rates. We get better with time, and we get better with training. So it really is time dependent. When you start out, there are risks because you're using a new piece of gear, a new component you've never worked with before. You've got to train people up. It's natural at the beginning to be, and one of the downsides to single use is there's not a lot of choice at bench scale, especially for upstream. And so you often work with traditional glass reactors on the bench and so on, and then it's hard to transition to a 50 or a 200 or a bigger bioreactor in manufacturing or pilot plant. It really is all about training. Oh, ultimately, yes. Ultimately, yes. Yeah, so the question is, the statement was made that companies who rely on stainless steel typically can achieve 95% success rates, and the principal risk is contamination or something like that. We get there too, and we run typically single-use bioreactors in the 50 to 1,000 liter range. And once you get over that initial issue of training, you can absolutely achieve that, because you're putting it on the manufacturer to create the product that you want, the component that you want, and then you're putting extra effort in training your operators to handle it properly. So there might be a slower start, a slower learning curve at the beginning. There's a lot more to worry about, but ultimately we get there and we're able to run consistency runs and produce a product. Supply, if I might just start off.
Repeat the question.
Yeah, so have we seen issues around supply chain where we're obviously very much dependent on the company to deliver what we need when we need it? It's not installed back there. We need it to come in, and we need it to come in on time. So we spend a fair amount of time, more than one would do if you had a fixed asset plant, in supplier management. So supplier quality management, supplier management in terms of product on the shelf and being able to get what we need. We buy stuff ahead of time. We make sure that we've got a good stock in the back room just in case something goes down. But we're friendly, really friendly, with our key suppliers. Absolutely. And if you, the advantage to us, we've worked around the fact that a gamma radiated component typically has a 2-year shelf life. So you can build that in.
Thank you very much. I appreciate that. We'll have time for some additional questions at the end.
All right, let's see if I can get this microphone to work. So, first of all, thanks for the opportunity, and thanks to all of you who are giving up your lunch hour to come and listen and have this great discussion. We'll continue with more points afterwards, so please be thinking of your questions. So, the other thing that I'm grateful for in being able to do this presentation is I got to know my colleague who lives in the office next door to me, Tom Pembino, who helped me put this together. He's sitting right over here. So I want to say thanks to him for helping a lot. We wanted to talk in context a little bit about a project that we have been working on at IPS for a few years, working through the DoD and working towards medical countermeasures, advanced development and manufacturing for medical countermeasures. Of course, it's a government job, so there's lots of letters. So this particular facility was built to make anything, anytime, any amount, and we don't know what it is yet. So the questions of how do you build a building like that can do any of these platforms, that can respond in less than 3 months from the time you know you need it to the time you make your first dose, became a challenge and really is the kind of project that's well suited for single use. So that's where we can talk about where single use does apply. Interestingly, when I got here this morning, Eric said, I wanted you to wrap up because when you talk, I want you to be able to stimulate some more discussion and conversation, and I know you'll challenge the audience. And I said, well, what does that mean about me? You think that I'm going to cause arguments? And he says, well, I know you, but actually it's because as a consultant, you see both ends of this business. You see why people pick stainless steel. You see why people pick single use. And help us understand that distinction and help us see what you see as trends. And so, interestingly, before I knew he wanted me to start arguments, we actually put together a presentation that's based on the concept that there are a lot of assertions out there, a lot of assumptions that people make about single use, and they're blanket statements that are true sometimes. And so the blankets need to be pulled back a little bit and talk about what's for real in the business. So that's how we're going to talk things. So the first one is, the bold assertion is that single use is faster. Always, right? So it is a rapid implementation. It is the right choice when you know that you're not going to have time for new process development after your clinical studies were done in single use. If you're not going to have that time, you can't get back into stainless steel. We know we have strategies now for faster filings. We know we have needs for faster filings. So we know a couple of companies that looked at Ebola and were in Ebola and had product ready to go in 3 to 6 months. And we have that with flu, we need that with pandemic, we need that for anthrax, we need that for all kinds of different response items. So we're looking at single use to help us in that space. But in reality, there are things that make this challenging, and single use isn't always the fastest answer. If you already have a stainless steel system up ready to go, you've got your CIP and SIP systems in place, you've already spent the money, it's actually pretty quick to get into stainless steel as well. So it's really, as I think Joanne mentioned, it's thinking hard about where is your investment, where are you in your manufacturing process, and what's your business case. The other next one that we talked about is single use is cheap, right? That's why we're all here, because it's low cost. So, and there certainly are things that are reductions in cost. Obviously, the capital investment can be pushed out because it's faster, so that's an actual savings from an NPV point of view. It's there's definitely a reduction in the size of the facility that you need, and you can push your costs out to know when you have product. So a lot of what we do in this business is pretty high risk, because I spent 20 years at Merck, as Eric mentioned, and we built 3 huge buildings to make Zostavax, Gardasil, and Rototec. 10 years we started building before the products were licensed. We were spending millions and over billions of dollars to get ready for those new product launches. This gives us that opportunity to push that out and spend that money when we actually have a product, when we know we have a need. And again, that's finally really important in some of these rapid response kind of areas. So, but the reality is, you take on a big cost. It was mentioned that if you do too many batches, all of a sudden it costs more to buy these big, complicated, expensive bags than it does to have bought a single-use or a reusable stainless steel in the first place. So the question is, you got to make that decision before you know. So how do you make that call? And it's sitting down with your marketing, sitting down with your regulatory people, and really trying to understand where your product is going. It's very much driven by your business decisions. So another bold assertion is single use is what facilitates open architecture. So now I can have ballrooms or dance floors, and I can make sure that I'm doing all my operations in one space, and I can routinely cross-train my operators, and they can run from the chromatography column back to the fermenter and turn on the fed batch, and then they can run back over to the TFF skid and pull off the peak, and then so but honestly, that starts to get challenging from an operational point of view. So the reality is, these are all complex operations. All the unit operations require a lot of training, and I think you heard everybody up here mention how important training is, and how the operations areas need to invest in single use. It's not free. So the advantages you get are real, but it's not for free. So another, the deviations. Something goes wrong, you get a leak in the room, or your environmental monitoring goes skewy, or you have a power outage. Now it impacts everything you've got going, every single batch and every single unit operation. So now there's a lot more unraveling to do to decide which of those products are impacted and what can actually make it out to the market. Internally, we still don't have full acceptance in large companies. A lot of times, although the external agencies are saying that they do accept this technology and this concept of a ballroom with closed processing when you can prove you're closed, there's still a lot of education and a lot of concern about that business risk around those products that you're putting through. Some of these products are millions of dollars sitting in that facility every single day, and that's actual product cost. And if you think about the revenue lost when you have a problem with these products, you shut down everything when you have a problem, you're really putting your business at risk. Bold assertion number 4, which is kind of a combination of all the above, is that single use is more flexible. And we built this facility in Florida to help make these products, and we believe that it does have a lot of flexibility. So, for example, we have DSP, USB modules. We have manufacturing modules. You can put in it what you want. Today you can put in it downstream, tomorrow you can put it upstream, day after that you can put both if you have a small process, and do them all in a ballroom kind of way. So we can do that. You certainly can't do that with stainless steel. You certainly can't, move the reactors around from one room to the other day after day. The way this facility is designed, we can run each of these modules separately. We can take the 2 left-hand and use them with the media buffer prep purple one in the middle. We can use the 2 middle ones if we need to segregate, and we want to keep those operations upstream and downstream separate from the rest of the building. So you can do that with single use. It's definitely there. Also, the idea of globally distributing your supply chain. So when we talk about vaccines, and Dani's talking about vaccines getting out to the markets, the people who need these vaccines are not all sitting around our manufacturing facilities. And a lot of these products are not very stable, and they don't do very well in shipping. So there's a driver to drop these manufacturing facilities all over the world. When it comes to a pandemic, 6-7 years ago, when we were in the middle of this pandemic response evaluation, the number 1 bullet on everybody's assumption list is all borders close. So if you don't have manufacturing for your vaccine inside your country, you're out, because nobody's going to be moving anything. So that means lots of small-scale facilities all over the place, as opposed to one big facility. And we certainly believe that single-use can drive that. But in reality, there's some constraints that come with that. The fact is that, the process is constrained to what's available from the innovators. So if your product really needs a certain ion exchange resin, and it's not available in a single-use configuration, it's just not available in a single-use configuration. You might be able to get somebody to pre-pack a column for you, but the time it takes to develop and put together a fully validatable package for a single use is going to extend probably beyond your needs. So you're going to be back to doing some reusable systems. Also, we talk a lot about small scale, and there's definitely a break in scale where this is applicable. Biosimilars are going to continue to be large scale. There's other kinds of biopharma products that are going to continue to be large scale. Vaccines are one of the ones that are amenable to small scale bioreactors. I mean, inherently, you saw Edward Jennings up there with a disposable bioreactor. That's called a roller bottle, right? We've been using them for hundreds of years. And, in terms of success rate and contamination rate, you open those bottles, thousands of them per lot, you open every single bottle at least 6 times in a manufacturing environment. So we've learned how to deal with disposable reactors, and we need to in vaccines. And carrying that forward really depends on scale and your product and your business model. And then the last bold assertion is that single use is equivalent from an operational point of view. So I love this quote from Tom. This is what we hear from our clients all the time. We don't need to worry about we're going to use single use. We want to do that. And we say, well, how do you think you're going to do this? How are you going to do your transfer from upstream to downstream? Oh, we'll address that later. How are you going to take your sample? Oh, we'll figure that out. So this is an issue that takes time and takes thought. So operationally, they can result in the same end product, in the same end quality, but there is a different journey along the way. And you need different kinds of training, you need different kinds of quality systems, you need different batch records. You need to think about, the tubing as it comes across and it lays on the floor. Is that really a quality problem? I'm going to guess your quality organization is going to tell you it is. But in reality, so you need to figure out how you're going to tie those up. How are you going to manage all of these connections and make sure that the right connection is made? And we were talking in the car on the way up here about RFID and the opportunity for RFID to tell you made the right connection at the right time. So that's the kind of tools we're still looking for to facilitate the operations. So that's really a summary of where we were, thinking that we wanted to start the conversation. So we glad to have more conversation, more challenges. To really think about when is the right time to use this technology. It's not the solution for everything, every time, for everybody. So well, thank you for your time, and I guess we'll have some more discussion.
Thank you, Sue. Thanks so much for that. I knew we could count on you to bring a little bit of provocative elements here. We have a little too much love in this industry, so it's good to have a little bit of controversy. We would like to have a couple of questions. I know Sue brought up a lot of points, so if we've got some time, we've got a little bit of time left, so question.
So what I meant there is that... Oh, yeah, sorry, I need to repeat the question. So what does the bullet mean, the less technical focus for operators? So there, if you're really thinking you're going to cross-train operators and move them from one side of the room to the other, I know in every facility I've ever been in, no matter how big or how small, tiny little university academic size labs, there's always an upstream guy and a downstream guy. So the conversation around ballrooms, and you get into the operational lean strategies, is, I can cross-train my upstream guy to go check and pull the peak off my downstream guy's column, or, something. And you lose that ability to really build expertise when you do that. And so it's a trade-off, obviously. If it's really a quick valve switch, can you count on that guy to run over there and switch the valve without really fundamentally understanding? Probably. But you're losing that value of building long-term expertise in any one operation.
Okay, thanks very much. Thank you again. So we have a couple of minutes for the panel. We've covered a lot of ground. We've got, as I said, we've got an exceptional brain trust here. We've hit on a number of issues that I think are controversial, and if some of them you'd like to explore a little more in depth, now is a good time. Okay, the question was, are you generally pleased with process performance of biobags? Is that?
I think in general we're able to get good performance in single-use bioreactors. Again, in our plant, we tend to be at the smaller scale. Sometimes there's demand for just phase one processing, so very smaller volumes. So we'll do that in some of our use the single-use bioreactor as a production reactor. But we normally use single-use bioreactors as in the seed train. I think I did mention that we have, at least in one case, there was an issue with compatibility with a leachable issue, and that's it's a very sensitive part of the process at that point. So you can impact the cell growth, and so we had to take some measures in terms of pre-treating the reactor and so forth. So it's nothing. With medium, so it's nothing you would not want to. That was a phase one process. That's nothing. You certainly wouldn't want to have to do that for a large scale, commercial process. And so if that did go forward, we would have to go back and work on that. So sometimes there's been those kind of workarounds that you have to do. And then the other issue, again, I think we talk about failure mode. Contamination. With the contamination with a stainless steel reactor, it's going to be because you had some component somewhere, you had ingress, or you didn't get something sanitized completely, or there was some component, some very small component failed. The failure mode is going to be different, obviously, in a single use. I mean, you can be pretty sure that the bag you get has been pre-sterilized, and we haven't had an issue with failure of gamma radiation. Where we do have issues, again, is in the areas in the interconnections, right, where you're connecting the bag to a sample line or to an outlet line or something like that, and it wasn't connected quite right. Or you're trying to steam it onto a stainless steel system, and you haven't accounted for the temperature differences, and you have some loosening and so forth. So, but overall, I think we have good performance, but there are some things you have to watch out for. And in at least one case, we did have an issue with a leachable situation.
Any other comments?
I'll just mention. In terms of, I think, questions revolves around robustness as well as performance. Is this guy working? I just have to get really close to it. So there are two aspects of this. This is performance in terms of mass transfer and being able to support the cells the same way that your stainless reactor does. But then there's also a question of robustness. And I've recently had a kind of incredible experience. Our partners at Genzyme are working on processes that are extended processes, semi-continuous. And they ran a single-use bioreactor, 50-liter scale, for 160 days straight, and only stopped because of the holidays. The bioreactor just kept going. So that gave us a lot of confidence in terms of the robustness of some of these components and the way that they're put together and the way that they're engineered. So they were working in the...
Can you repeat the question?
Oh, yeah, sorry. They were working in... so the question is what cell density they were working with. So this is a CHO line. They've also done it with 293s. And they're working in the 20 to 30 million range.
And we have time for one more question?
Yeah.
The question is how many manipulations during
160 days? Yeah, so what they were doing is it's kind of fancy perfusion. So a continuous feed, fresh media. They have a custom designed autosampler port that periodically pulls out samples so they don't have to keep penetrating the bag. It's all fixed in place and it just does its thing. Some fancy Raman spectroscopy for monitoring metabolites and viable cell density and stuff. They really, in terms of the process design, they spend a lot of time engineering out all of those risky manipulations as much as they could.
Okay. We have time for one last question.
Yeah. Well,
the question is, can you repeat the question? It was in regard to continuous processing and downstream hardware connectivity? Downstream processing and continuous processing and downstream hardware connectivity. Was that the issue? Single use.
I don't want to dominate the conversation here, but we spend a lot of time sorting out the connectivity issue. When we first started with our smallpox vaccine, we were in a really big hurry. 9/11 happened, and we all of a sudden had to get going, like yesterday. And so we assessed technology-based really on availability and could we get it in and could we get the operators to train up on it. We had tubing welders coming out of our ears to try and figure out which one was the most robust and so on and so forth. Things have gotten a lot better in that there are a lot of new vendors out there with aseptic connectors and so on. You don't have to rely on the tubing welders so much. The more you move to those kinds of built-in solutions, the better you are. We spent a lot of time building little manifolds with a valve on the end and a piece of tubing that we could weld on and autoclaving it and steaming it on, and it got pretty hairy. We pulled it off, but it's not the way you'd want to start today. There's a whole lot of different solutions out there. Companies coming onto the market all the time with new ideas and new solutions. So your choices are a lot better now.
Okay, I'd like to thank our audience. I'd like to thank Brian Kane, and I'd like to thank BioProcess International. Thanks for this meeting. And thank our panel. Absolutely. Thank our panel for all the time and energy.